Welcome to this ISFP sponsored dialogue series. Myself, Dr. Naresh Ranga, and I welcome today's guest speaker, Dr. Akwasi Boadu. So, let me introduce to you, sir. So, you are a alumni of University of Kuala Zulu Natal, Wesbury campus, College of Health Sciences, University of South Africa. You have 15 plus years of experience with education management and teaching, including being a chief invigilator in the government public examinations. And your research areas are natural products chemistry, computational chemistry, etc. And presently, you are holding the post of director cum principal at Wesbury College of Science, Port Shepstone, South Africa. So, with this, I welcome you, sir, to deliver your talk on the topic key concepts in natural products chemistry. Over to you, sir.
Thank you. Thank you. Thank you, Dr. Naresh, the coordinator of this lecture series, for inviting me to the IQAC ISFCP lecture series. I would also like to extend my sincere thanks to the chairman of the college, S.H. Praveen Garg, and also to the director, cum principal, Dr. G.D. Gupta, and also to the vice principal, Dr. R.K. Neren. Thank you for inviting me to this series lecture. Today, in this lecture series, I am going to be discussing many important roles played by natural product chemistry in drug discovery. I would like to also greet all viewers and colleagues, listeners, for tuning in to this lecture. And I am very grateful that you can spend 20 minutes to 30 minutes of your time to listen to such a lecture. My lecture today will be on key concepts in natural product chemistry, whereby I'll then narrow it down into plant material. Look, chemistry is life, and life is chemistry. Even when people fall in love, they say they've got chemistry. Means that chemistry is everything. But how do we use chemistry? How do we let chemistry work for us? As natural product chemists, I would want to navigate with you through some processes that we take as natural product chemists in order for us to play an important role in drug discovery. From slide 2, we can indicate, as we can see, that chemistry is the science of the composition, structure, properties, and reaction of matter. Life itself is full of chemistry. The understanding of disease and treatments stems from the research in chemistry. Natural products have contributed significantly to therapeutic products in some pharmaceutical industries as the primary driver of drug discovery. With all of these things that natural products have done, we have seen a decline or a scale down in big pharmaceutical companies in their research into new therapeutics from natural products because of the complexities and the cumbersomeness of the metabolites in isolating, or first in extracting, identifying, and isolating active compounds. But as natural product chemists and as pharmaceutical chemists, we need to revitalize the natural product chemistry in drug discovery. But to revitalize it, it will depend on an improved and efficient methods of extraction, identification, isolation, and structural elucidation combined with biological activity. So you can see in slide 3 and slide 4, these are some of the contributions that natural products have made in medicine or in the pharmaceutical industry. In the 1800s, morphine was discovered from opium poppies. We can see that there is a whole penicillin and all that has been discovered. Even during COVID-19, on slide 4, these are products that have been discovered from marine to terrestrial to plant or even fungi. We are finding solutions to diseases. We are going to look at other things such as identification and authentication of natural products. For you to be able to research into natural products, you need to identify the natural product, authenticate it. How do we do that? We do this through 3 main approaches. The first one is the random approach. This random approach is a random selection, as the name says. It's a random selection of natural products from a particular geographical location for screening of targeted phytochemicals, and bioassays are performed. In the random approach, this method has no prior pharmacological action or no prior pharmacological history. Therefore, we use something we call a sequential extraction. During this random approach, we pick a plant or any natural product, you extract whichever part that you want to do, you get the crude extract, then from there, you do a preliminary phytochemical screening. In so doing, you are able to identify the class of phytochemicals that are in those plants. You then do a random bioassay to check which extracts or which fractions have got any activity on what particular protein target. The second approach in identification and authentication is the ethnopharmacological. I always like to use ethnopharmacological, but others will say ethnobotanical survey, others will say ethnomedicinal. But I believe that ethnopharmacological captures everything, all natural product, whether plant, animals, fungi, or any other thing. This ethnopharmacological approach is an oral tradition that has been passed on from generation to generation. And as such, it uses crude extract in the community to manage diseases. This, we know the pharmacological activity of that part of the plant or that part of the natural product. And therefore, we are able to do something called a parallel extraction and tests for the isolated compounds. In this particular approach, we would find out that although we can get the isolated pure compound, sometimes it does not work because some of the phytochemicals those are secondary metabolites, some of them work in synergy. And therefore, when they work in synergy, they cannot work alone as a pure compound, and therefore we don't get results from it. We then look at the modern type of ethnopharmacological approach, which is what we call the traditional medicine system. The traditional medicine system is similar to the ethnopharmacological approach, but this method here is more modern, is newer, and is well documented. This codified method of well document of traditional natural products are mostly practiced in Asia, especially in India. Its advantages over ethnopharmacological are that it is based on very good empirical practices, well-developed pharmaceutical formulation, and is much more institutionalized. Therefore, there are more records, written records, than ethnopharmacological. Remember, word of mouth, it changes through interpretation. But when something is documented, it can be used properly. With this, we will then move on from the identification and authentication of the natural product material, and then move on to the crust of the matter. How do we extract? How do we isolate? How do we purify? What are some of the mechanisms that we need to use in doing these things? Now, there are two types of extraction, parallel and sequential. I'll move on to slide the next slide that I've spoken about. I have spoken about the random ethnopharmacological and all these approaches. Now I am narrowing down to plant extract. I will then indicate the parallel approach of extraction. The crude plant material is then extracted through certain means. It could be decoction, infusion, maceration, and or even succulent extraction or microwave. All of these things depend on the biological activity, or depend on the heat liability of the phytochemicals. Some of it also depend choosing the particular extractive method depends on the ethnopharmacological practices. Mostly polar substances are used, like water, ethanol, or a combination of them. Therefore, most pharmaceutical chemists or natural product chemists use some of these methods. Some of the solvents are hexane, that are non-polar, hexane, diethyl ether, or we can even have something like ethyl acetate and the rest. So we use these solvents in order to extract the secondary metabolites from the natural product, in order for us to be able to analyze them. Therefore, when we look at the parallel approach to the assay-guided isolation, we extract with solvent, either from 100% and a combination of it. After that, we find fractions. These fractions are found either through chromatographic methods, TLC, thin layer chromatography, column chromatography, such that you get fractions of these crude extracts. Now, by obtaining fractions of these crude extracts, you can either go and do a biological activity. Then you can fractionate further, or you can isolate the active compound and purify. Other times, we don't do any biological activity. After fractionation, we then try as much as possible to isolate all the active components, and then what we do is that we purify it. By purifying it, I will then go to the next slide, whereby we then have got to our parallel approach. Sequential approach also follows the same thing, the same processes, but in our characterization and structural elucidation, after we have obtained the pure compound, we then go through processes such as TLCs. We then look at chromatographic and spectrometric methods. These methods are time consuming, but these methods are very important, such as your HPLC, your LCMS, and your GC. Your NMR is one of the most important aspects of the identification and structural elucidation of a pure compound. NMR provides many advantages over other methods, such as the ability of it to directly observe chemical compounds and target biomolecules. NMR also gives the basic skeleton of the pure compound that we've isolated. Therefore, NMR combined with FTIR, which then gives the functional group of the isolated compound, let us able to then identify the type of compound, pure compound that we've isolated. We've got the X-ray, that is if it is crystals, and all that. These are the methods that we use in then isolating and identifying pure compounds. Now, when these pure compounds are then identified, what then? Either we go and do biological activity, in vitro, in vivo, in silico, and clinical trials. Recently, most companies, big pharmaceutical companies, start with in silico. That is your molecular modeling approach, whereby we screen the compound for any activity. This in silico method is less cumbersome, more effective, it is cost effective, and give results in few minutes, if not seconds. Therefore, it is a go-to identification of biological process for most companies, so that we don't waste money and time on all the other things, and we know whether this will work or not. During the in silico methods, we found out that we could look at the absorption metabolism, that is your AMDET, and we would know whether this compound will work or not. In silico, if the compound does not work, or even if it works, we can create a pharmacophore which will actually target, which will indicate which part of even the pure compound is the active part that causes the effect on this biological or protein target. In silico, we can also then create pharmacophores, yes, I've spoken about it, but analogs, which can then be synthesized in the laboratory just to reduce the toxicity or any other side effect that this main compound may have. In sum, I would conclude by saying that natural product chemistry is evolving at a very fast pace. And now with machine learning algorithms and artificial intelligence, natural product chemistry are opening up a lot of opportunities in the discovery of more efficient methods, more candidates, more drug candidates, and also by also using it to identify repurposed medicines or therapeutics to treat emerging and re-emerging diseases. Science is around us. Let us look around us. Nature has given us all that we need, and nature is chemistry. I thank you.
Thank you, Dr. Akwasi, sir, for this wonderful talk. So it was a mind-opening session, sir. So on behalf of IQAC ISFCP, sir, I extend a really hearty vote of thanks to today's expert speaker, Dr. Akwasi Boadu, who spared time from his busy schedule to grace this event. So today we had an opportunity to hear your views on the topic, key concepts in natural products chemistry. And this will surely be going to encourage us in our future research events, sir. So once again, thank you for this talk.
Thank you, Dr. Naresh, and thank you to your sponsors, and thank you to everybody who made this happen. I also thank the staff of Wesbury College of Science for making this a success. Thank you.